Welcome everyone to our ICARE case conference today. So I wanted to jump into the case and then have our guest expert lecture or guest expert talk right afterwards to put it into context. So this is a patient that I had seen. The genetic testing had been ordered at an outside institution, and really the patient was referred to me for a discussion of her TP53 positive result. And she was a 35-year-old female with left breast cancer, early stage. It was ER/PR positive, HER2 negative. She had been diagnosed at age 34, and she was status post bilateral mastectomy and was currently on anti-hormonal treatment, specifically tamoxifen. So here you see the family history, which is a fairly extensive family history. So my patient is the 35-year-old in red in the bottom part of the pedigree. And then you can see here she had tested positive, but there are multiple family members that had been tested, including her brother who was tested positive, sister tested negative, her father was positive, and her aunt here was positive as well. And then you can see an extensive family history beyond these individuals. So I actually saw the father as well. So he was a 62-year-old male, self-referred to discuss the pathogenic variant in the TP53 gene. This was following me seeing the daughter, and the father had not been getting any ongoing management. So I suggested that he could come to our clinic for additional management guidance. He had been diagnosed with Gleason 8 prostate cancer at the age of 53. He was status post radiation treatment, radioactive pellets, anti-hormone therapies per the patient's report. I also saw the aunt again after I had seen the original proban, and she is a 53-year-old female, self-referred, seeking surveillance recommendations. And she was, again, tested by her local provider after her niece, so meaning the original program I was talking about, and her brother were tested positive. She was sent for a dermatological exam and identified with squamous cell carcinoma. And a colonoscopy in 2018 showed 2 hyperplastic polyps, and this had predated any knowledge of having this TP53 mutation. So again, something that needs follow-up. So here are the genetic test results. And again, this is interesting because you can see they were done by different labs. So for the original proban, the 35-year-old, the testing was done through Ambry. And the result, you can see here, TP53C 541C to T, and it was classified as likely pathogenic. So for her dad, this testing, I believe, was done at Invitae, and this was classified as straight pathogenic. And then in the proban's aunt, initially when she had the testing, it was done through ARUP laboratories, and the result was likely pathogenic. And then after the local oncologist who she had seen saw this result, they ordered the XG testing. It wasn't tumor testing. It was the germline testing through Tempus. And that came up as a variant of uncertain significance through, which is the GeneDx report. So you see this variety of test results in this family for the same exact variant. So I went to CanVar, and again, you can see here the depth of information we can get through CanVar. You can't see it here, but I pulled in the interpretations across the various labs. And again, you can see that this has been seen across various labs. If you could see the screen behind the CanVar, you'd be able to see that, again, it's all across the board in terms of how this is being interpreted, just like we saw in our experience with this family. And then you can see here with the genetic test results, again, this is the resource that Dr. Turnbull is going to be going through with you over the course of this case conference. But again, with this specific variant, what I find really useful if I use CanVar to go into ClinVar is you can see that on the home screen, you can actually, there's a link directly to ClinVar. On top of that, if you can read here, again, it's hard to read. It says right away, ClinVar status, conflicting interpretations, again, which is useful. And then you can see the ClinVar interpretation here, where, again, you see clearly conflicting interpretation of pathogenicity, one lab listed as pathogenic, two likely pathogenic, and one uncertain significance. Again, I think some of this is our specific family where they got tested at multiple different labs. So I actually did go back to the labs when I saw the ant because of this VUS result. And I wanted to find out if RNA would be useful. And again, the bottom line there was that it would not be helpful with this missense variant. And I think there is some evidence of pathogenicity, which is why various labs are calling it pathogenic or likely pathogenic. What their additional insight was is this may not be a typical Li-Fraumeni syndrome mutation, but does lead to some increased risk. And again, the family history is not classic for Li-Fraumeni syndrome. With Invitae, again, they gave us the specific way that they had classified, which I always find useful. So basically, they said that this variant has 6 pathogenic points in their variant classification system, which they call Sherlock, with 4 pathogenic points needed for likely pathogenic and 5 for pathogenic. And there were 0 benign points assigned to this variant. So looking at medical management, this could be a reduced penetrance variant. But again, at this point in time, we don't really have specific management guidelines to reduce what we do with these patients who we screen for Li-Fraumeni syndrome. So we recommend the annual brain and total body MRI, follow up with the local gastroenterologist with colonoscopy, initiation of endoscopy, annual dermatology exam, and then comprehensive physical exam. Now, implications for the proband's children, both the son and the daughter, who are both minors, were tested and both tested negative. So follow-up is, I'm planning to see the family back in about a year. So with that, we're gonna come back to this case, but I wanted to introduce Dr. Turnbull and also have her give you more of a presentation on this amazing resource, CanVar, that they've developed in the UK. So by way of introduction, Dr. Clare Turnbull is the Health England Professor of Translational Cancer Genetics at the ICARE. She's also an NHS consultant in clinical cancer genetics at the Royal Marsden NHS Foundation Trust, and a consultant in public health medicine for Public
Health England.
Okay, yeah, I thought there was a word.
You've given me some kind of eponymous chair that I don't have. Which sounds very important. I'm like the professor of the whole health of England. It's quite a weight on my shoulders, you might imagine. Right, thank you very much, Toya. I'll give a quick whiz through the sort of background and then show you the platform a little bit. So we're reasonably cohesive in the UK around cancer susceptibility genetics, and we put a case to Cancer Research UK. They had a kind of funding stream called a Catalyst Award, which was to sort of promote rapid change in an area of health-related research. So we put forward an application to develop data resources, clinical and educational tools to leverage cancer susceptibility genetics for early detection and prevention of cancer. And there were 6 work packages put forward, and one of them was around variant data resources that I am leading. There are other work packages. Mark Tiskovich, whose name we've already heard today, is leading on clinical guidance with Helen Hansen, Diana Eccles on patient tools, and so forth. So, and the sort of the notion was this was to fund activity which wasn't classically, pure research, but was needed to deliver cancer susceptibility with genetics within the healthcare service, and the kind of manpower that you don't have right in the clinical service to fund that. So there were 4 bits in the work package around variant interpretation. We wanted to establish a national laboratory network with a special sort of dedicated meeting. We wanted to create a resource for variant interpretation that supported this network. So some kind of national MDT platform. Thirdly, focused analyses to better understand variation, and fourthly, to support international collaborative efforts. So in regard of the group, CanVar-UK, Cancer Variant Interpretation Group UK, we'd actually initiated it probably about 18 months earlier, and that just the grant gave us funding to really support it and grow it. We've got 267 members who attend regularly. We have monthly meetings, and between the monthly meetings, we have a very strong sort of network for communication. We're about half, this poll is actually very old, but half clinical scientists, half clinical geneticists, and most people work exclusively or predominantly in cancer susceptibility genetics. About a third of people are broader than that. Sorry, yes. So yeah, so we have in our monthly meeting, we send out a variant ahead of time, and we give some case information where it's relevant. And then the clinical scientists in the 18 labs across England who are part of the network. So 18 labs across England, 4 in Scotland, 1 in Wales, 1 in Northern Ireland, all represented in our group. So at least 1 person from each lab tries to feed back on the variant and do a classification. And in part, that's to sort of reach a consensus, and in part, it's around education and improving consistency. So we have a little tool whereby they feed back their different scoring for different criteria, and then we can integrate those, but we can see what their final classification was for where they gave their points. And people tend to write lots of notes. And then Alice, whose photo is there, pulls all this together, and then we can sort of use it. And it's very good around getting insight and trying to get more consistent. So ACGS is the national body for the clinical diagnostic scientists. So until 2020, they did an updated, quite broad specification of the ACMG guidance in much more detail. And then actually we in CanVar, there are quite a few elements that were more specific to cancer susceptibility. So we published some guidance consistent with the ACGS, but specifying particular things around cancer susceptibility. So a bit more around PS4, that type of thing. And then also we sort of, ahead of Sean Tavtijan's publication, pulled together some recommendations about using the point system and combining evidence and different categories that weren't in the original ACMG. So we looked at all of the sort of, at that point, sort of specific guidance that was available, and the different numbers of points you could get, and had a look at which combinations you could generate. And so again, it was some sort of updated guidance for CanVar around sanctioning use of different combinations of evidence items beyond the ACMG. And we also sort of, again, these sort of scorings within the bus category, which again, there's this sort of descriptor, which is very useful. It's not good for patients, not good for mainstream clinicians, but actually quite helpful within our sort of genetics discussion about a hot bus versus an ice cold bus. And that's actually, quite important in how we're classifying them in terms of the sort of clinical reporting, and also how they're stored and so forth. We also published reasonably explicitly on combinations of evidence items that shouldn't be combined. And we have put that into a grid. So that's now available. You can see on our CanVig part of our website, with quite specific didactic rules of, so red you can't combine, pale green you can, and the bright green is a sort of caveated combination, and the grey is not possible. So we've done that for pathogenic and benign, and these have evolved. So those sort of versions of those broad CanVig guidance held on our website that we do keep up to date. So it's as bits come along, as new publications come along, we ratify them in the CanVig group and update. Something that this is IMPRESS in genetics and medicine. Again, a framework around variant reclassification and recognizing limited resource in the labs, that they can't be classifying and reclassifying and up classifying and down classifying all the time. So it's actually some sort of practical written guidance following a series of national meetings around expectations and responsibilities around variant reclassification and how that's communicated within our national network. And we did sort of particularly identify that there are these variants that would sit at the kind of low end of likely pathogenic or the hot VUS end, and actually particularly if they had only just been sort of gone up or down by a small number of points, that these were had a sort of category of being potentially changeable and the actionability threshold, and actually that we issued some specific caution about a pause before you go out and contact all the retrospective patients. And again, it's all sort of fairly sort of straightforward, obvious things but laying them in paper, that we have some documented, recommended, consistent practice in how we address this, we felt was useful as a community. We also have some interim gene-specific guidance, so monitoring, sort of waiting for the formal mismatch repair BRCA guidance. We've also got some SDH guidance, which there isn't an SDH VSIP, but again, sort of waiting till these groups publish and just sort of keeping a document that, again, we can be nationally consistent and agree what we do for these genes while we await the formal release of the final VSIP guidance. And another thing, so this is Miranda, who's one of the, she's a senior clinical scientist in Sheffield, has again drafted a series of report wording templates for different scenarios. So a male report for an ATM variant, pathogenic variant, a report, VUS reports for high penetrance ATM variants, reports for reduced penetrance BRCA1 variants. So again, that we are nationally using consistent language in how we report these variants and what we recommend. I'm going to give you a demo of this in a moment, but this is the second element, is our platform, CanVar-UK.org, and just to acknowledge Jankut and now Subin, who have worked on the development of this. And again, this is, as I said, it's a national multidisciplinary team tool, so it's got resources in it, but also tools that people requested. So this is our PM5 tool, which is a joy because it pulls out all of the base substitutions at the codon and gives you a live snapshot of their ClinVar status and their Revel score. And again, so our recommendation for PM5 is around using the Revel score to inform it, and that's actually very useful. And as Toya said, actually trying to find your variant in ClinVar can be, if going in directly, a bit tricky. So this links in live with ClinVar, and gets an up-to-date classification. Again, with the community, add in silico tools as people request them. Generate a Google Scholar search of a quite tight defined but flexible variant nomenclature. And this, again, the different data sets from Nomad that obviously you want access. The default is the non-cancer Nomad. And again, people fall into that trap of using normal Nomad with TCGA. But we also have non-cancer female, which is the appropriate group in some circumstances to use if your reference point is, for example, PS4, thinking about female cancers. And then we have started a program about 5 years ago of all the laboratories. This is in England now, 18 laboratories in England submitting all of their historic testing data and the variants detected. And then these are, so if you're logged in as a CanVig NHS user, you can see the breakdown, but actually as a non logged in, you can see the total. And as it's not of that much interest where they were detected. So we've got a numerator and a denominator for the number of times events observed in total, and then in people of self-reported white ethnicity, which they hold within the cancer registry. And there's various issues about small numbers, so they won't let us report any other ethnicities on a public database because they are too small numbers. So there's a little bit of a battle. These data are submitted to Public Health England and NHS England, which and they're pseudonymized, so the patients aren't consented. So there's various guards around uses of the data, but this does allow you to, in comparison to Nomad, then do a PS4 Fisher's exact using the white-only data, bearing in mind that this is an enriched population who are having BRCA testing. So, and what's nice about these data, we're just getting to grips with them. They're messy, they're imperfect, but they're individual levels, so we can collapse them and use them for PS4, but we can also, on an individual level, they're submitted with their NHS number, which is a unique national identifier. So we can then link them to the cancer registry to get cancer diagnoses, but it also contains data on chemotherapy, radiotherapy. The labs also submit somatic testing data. It may be different labs, hospital episodes, which has endoscopies and so forth, death and prescription data. So colleagues Mark Tiskovich, Antonis Antoniou, Paul Ferro are part of this program. So they're doing analyses. And then a couple of members of my team are looking at linked data on MMR. So that's a slight diversion, but tenuously relevant. And then as our MDM tool, what's very nice is people registering not just their provisional classification, but also some notes. And then they can send out to, they can just document their notes. They can send out a standard email, or they can send out an urgent email. And so then you get an email to the whole CanVig network, which has the variant in the title of the email, or urgent with the variant name if it's there, people are worried about a reclassification or something very important. And then if I click on here, it takes me straight to the variant on the black button, the appropriate page within CanVar, so I can type in my response, and then all of that sort of is documented and captured. And prior to this function, which Subin introduced maybe in October, just the wealth of discussions that just in emails about variants, it's almost impossible to go back and find. So this is really useful to just sort of have these logged here. But this is only if you're logged in. So this is only for the NHS CanVig users. And it's sort of probably time to re-explore, but people felt that they wanted that protection around, I mean, no one's mentioning patient names, but just around clinical details and also their opinions and around the variant classification. And then there's a gene-specific page as well. So this contains useful things like the exon boundaries and sizes and intron lengths, so you can sort of see if deletions are in frame and that kind of thing. And then also our sort of gene-specific guidance So this is for BRCA1, so it's quite detailed, but just for other genes, sometimes, you know, just things like, you know, the most 5' base that's known to escape nonsense mediated decay, or there's some key details. So people, so this has got limited access, so only particular people can use this and update if it's a sort of CanVig approved entry. But again, just a resource for gene level guidance rather than variant level. So CanVig, we submit to ClinVar when there's a consensus CanVig classification, and again trying to a detailed description of how we got to our conclusion. All of our resources, our guidance, meeting recordings, slide sets from our meetings and so forth are stored on our website and are available. We've got a group, so there's 7 senior clinical scientists, so we also meet in between times once a month. It's a really useful group to just flesh over those report wordings or the recommendations and so forth, because the CanVig meeting is a very large meeting. It's not one for sort of detailed debate. And then we share a sort of provisional version of things with the CanVig group in the monthly meeting. And then just a few, as I said, the third element of what we do is spin some genes, some sort of active analyses to address questions. So Jean-Cout in my team did a big analysis looking at 5 curated sets of massive functional assay data, MAVE data, and 44 in silico tools that we, and also looking at them in combination and so forth, and then looking at the outputs in terms of likelihood ratios. So these again would slot into that points-based system. Lucy and my team looked at PM5, and again using that Findlay data and the MSH2 functional assay, again curated, to sort of quantify across a whole gene in an informed fashion actually how much was concordance of if there was another base substitution at the codon, how much is that actually telling you by using a large data set, quantifying that, and then whether your observed variant is more deleterious on different tools, whether there were 2 pathogenic variants at the codon and so forth. So actually, and then quantifying these in terms of likelihood ratios, which then slot into that exponential ACMG scheme. And then a big analysis of SDH genes, and really trying to quantify how much it's telling you if you see a very rare variant in an individual with a very specific phenotype, and also quantifying some subphenotypic features. So again, as likelihood ratios that can be converted into those exponent points within the ACMG numeric system. So again, just going alongside CanVig. So I think that's all I was going to say. Oh, actually, can I share my screen now? Sorry, because I just want to go to CanVig. So it's CanVar-UK.org. So I was just having a look at that TP53 variant that Toya was showing us. So I'm just going to look up another variant here. So let me go to, let's look at that BRCA1 variant. So we just type it in C.5207. So it will start completing for me. And then I want the T to C. There we go. So yeah, so you get your front page, so various useful information, some classifications down here. You can go to ClinVar, it will load ClinVar and take you to the page, but it pulls in the classification, as I said, this PM5 function that will then tell you about all the different base substitutions at the codon. It takes a few moments because it has to go and retrieve them all from ClinVar to get a live classification. You can go to Bracket Exchange, you can go to Varsome, which is a nice site, particularly if you want to look at the effect of stop codon or whatever. LOVD, and then, as I said, the Google Scholar search, it opens a new tab and does that nice appropriate search term. And Google Scholar goes to Google. You can do Google as well, but that's far less specific. Your in silico predictions. So again, I've sort of highlighted the ones that tend to be most useful. Revel is very much in silico tool du jour in the UK, along with Splice AI, Maxent is there, your domains and so forth, and then Blossom scores and conservation there. As I said, this case control frequency here with the numerators and denominators in total down there, your Nomad. We've also got the 1,000 genomes project with the different subgroups, but this variant isn't observed in here. Various Gen Epi papers. So this is the original Eastern one. So this is in here. And then it's explained, but we give the published log likelihood ratio. And then we also give an ACMG log likelihood ratio, which is slightly, it's to a different base. So this will make sense if in line with the new Enigma VSIP guidance, but converting the evidence to exponent points to the correct base for the ACMG. Various selected splicing assays are in here. So these are ones that we know have been validated and can be used within the ACMG at an appropriate number of points with the various results. And then there's various published splicing assays, but actually Wessex is the main lab in the UK, so they've shared their splicing data, which again is just useful so people don't repeat the splicing analysis. And then, as I said, this is the page where people, so I could put my proposed classification, and I can upload slides. So someone's uploaded the CanVig discussion summary, but also if I had any of my own data, I could put that. I can make some notes, and then I can just save it, or I can email around if I wanted to ask a question, and then hit post. And then there's this gene, as I said, this gene level information, where again it's just all of the different sort of specific guidance around classification, but also quite usefully information about the exons and the bases and so forth. So is there, oh yes, oh, and then the other thing, what else? If I want to download it, but so Toya was saying this, so again, this just pulls out all the data that's in there, the variant report, but it means you can just download it as a PDF. So again, depending on what your use case is, it's just a way of pulling out all the in silico predictions, some of those Gen Epi studies, the functional studies, and so forth. So just that's downloadable. So I think, let me just click to search another variant. So I'm just going to look at an MLH1 variant. So we've got, I think it's about 100 genes in there at the moment, but Subin's just in process of extending to include some more, particularly some hem-onc genes. So we've had a big push on germline hem-onc activity in the UK, not least because they're all getting whole genome sequencing, or the AMLs are. So there's quite a lot of myeloid variant interpretation going down. So so yes, so this is an MSH1 variant, and again, just that, again, that so for these ones you can go nicely straight through to your insight page and find the variant in question, which is useful. And then that's the old insight paper, and again, but that would take you to the insight page there. And there are this is MLH1. There are some assays in here, but clearly no data for this variant. I don't think I have anything much more to show you. So maybe Toya, shall I hand back to you?
Yeah, no. So what we wanted to do is really give people an opportunity to ask questions, and I'm gonna so I was amazed with what you all have built. I only became familiar with it because I was talking to Mark Tishkowitz when we were at the ACMG meeting several weeks ago. And he just showed us this tool. And again, many of us in the US use ClinVar, but I found it useful to use CanVar as a way to segue into ClinVar still. But then I get all this extra information, which is, you know, really fantastic. But I had, you know, some of the glitches that I have experienced is when I have, you know, you put your C designation, but then you have to put a bracket beside it, right? If you don't have the P, or it doesn't work.
You should just be able to, it should search. So if it should find, so I'm still sharing, aren't I?
You are still sharing.
Yeah.
So
okay.
What I've had to do sometimes is trick the system into putting two brackets when I don't know what the P is.
Oh, I see. So, but it should narrow down.
It does sometimes, but it doesn't always.
It might be.
I'm just like, yeah. Then, you know, just so people can use it, like these are just things as a new user I came across. The other question I had for you, and you kind of alluded to it, is so you guys are sharing consistently with ClinVar. So the variants you are identifying and interpreting in the UK should already be in ClinVar. Is that right?
There's a bit of a lag, and we upload to ClinVar the ones that we have discussed together in the meeting, the CanVig meeting, or that we've had a kind of email thread or discussion or whatever during the month. So it's not that we've reached effectively a sort of a consensus decision on. Yeah.
Right.
So it's not sort of every variant that's being classified in every lab.
Yeah. So my other question is, you again alluded to potentially opening this up, because in the US, we don't yes, we will, the way it works in terms of our workflow, or at least my workflow, but I think it's also what many of my colleagues do is we'll, you know, send emails literally to the labs as well as to our colleagues who are knowledgeable in variant interpretation when we get something confusing or complicated. Whereas you guys, it's amazing because this is a centralized system where you have clinicians and variant experts on the same platform trying to come to a consensus, which I'm not going to say we generally have, because even across the US labs, there's often broad ranges of how something can be classified, as you saw in my case.
Yeah, so, I mean, the other thing is it's often a sense check if anyone's got any cases or families. So particularly for Lynch, just going, we've seen this, it's in a proband, you know, 45-year-old with bowel cancer. Anyone else seen it? And also what the family history or the tumor findings is. So it's partly just flushing out if anyone else has got any additional phenotype or case or phenotype data that would inform the classification, and then also sometimes sort of a bit of toing and froing on, as you say, the specifics of how you would award points and classify it.
So is there any movement towards making it more open?
Well, I think that as ever, it's a sort of
Mark told me to ask you that.
Yeah, I think there's a split in the field, and you end up going along the conservative line. But maybe I can see Mark's here. Maybe it's time to ask people again.
I mean, I don't think there's as much awareness about this amazing resource, which, you know, again, I was thrilled that you agreed to present, because even without being able to get to the login only screen, there's a wealth of information here that's super easy to find that would make our workflow much easier. But if we were able to get into the secure part and, you know, ask about variants or tell people when we've seen variants,
yeah,
that would be fantastic.
It is. I mean, it is I'm always sort of mindful of duplicating effort, but it is doing something quite different to ClinVar. And I think as long as we submit to ClinVar and we access ClinVar and you use that information, it is a sort of collaborative multi-center platform. So yeah, so the only bits you're not getting are those lab specific breakdowns, which are just a bit more useful internally, because you can see then which lab has seen it already, and that those numbers we have to get sort of updates. They're always slightly behind, but it does give you an indication of you know which labs definitely have seen it. So you don't get that bit, and then yeah, at the moment the conversation and the classifications. But yeah, well, let's ask the CanVar group again, because it's one of those things. I don't want to discourage the UK users from using it.
Right. No, it's a fine line. I think everyone gets it. You want to have your own safe space to be able to have these discussions.
Yeah, but equally there's a benefit to everyone if the more people you consult, the more useful information you'll get, and that's a two-way street.
Right.
Yeah.
And your thoughts on my first case, the TP53?
So I have some broader thoughts on TP53 that the VSIP are way too punitive in terms of, so the VSIP guidance in terms of how you get points for case data. And they justified this. They said in the States, there's so much TP53 data that we need to be really careful, because it's being tested for in, you know, pretty much unselected breast cancer and so forth. But actually the whole ACMG is based on a prior probability of 10% that you will have a pathogenic variant when you walk through the variant classification door. So actually what you need to do, if you're testing genes either in you know, completely without phenotype or with very mild phenotype, you should give some minus points, or you know, some kind of way of adjustment. And then with P53, you know, if they've got early onset breast cancer and 3 sarcomas in the family and an you know, adrenocortical, that you should get many more points for those really heavy Lee-Frau-Many families, because at the moment the classification system is a bit frugal in terms of awarding any number of points. So unless you have really good functional data, it's really difficult to get a variant over the line for P53.
But can I ask you that, but it's two separate concepts in my mind, right? Which some of the labs treat in different ways. One issue is penetrance. The other issue is pathogenicity, right? And they're different. And well, so sometimes people are calling reduced penetrance, like we have examples of this in CHEK2, where some labs call a reduced penetrance CHEK2 variant a VUS because of reduced penetrance. So just keeping those separate is also.
Yeah, I mean, well, they are, but the problem is if it's reduced penetrance, its ability to score for most of the categories by which you get points for pathogenicity is impaired. So you're not going to get a really strong family history, and you're not going to get, you know, quite often the functional assay will be intermediate or whatever. So it's a penetrance and pathogenicity are sort of manifestations of the same thing. If you think that basically every variant has an effect size, and we would arbitrarily, you know, we tend to think of pathogenic being around some sort of high penetrance, and therefore for that gene, if the penetrance is substantially less, i.e. the effect size is substantially less, it's still disease associated. Is it pathogenic? It's, you know, you may have case control data that shows you it's very clearly disease associated, but it's probably not going to, it's not the same entity as a high penetrance variant. And when do you get down to like a SNP, a kind of GWAS SNP, which has an odds ratio of 1.1? So there is a spectrum, and we you know, for the ACMG was really designed for things that are high penetrance Mendelian. And as you start trying to apply it for things that are either lower penetrance genes or lower penetrance variants in high penetrance genes, it's very difficult to actually get those scores. So you're really kind of reliant on, usually for the lower penetrance things, for clear case control data, aren't you? And then in cases like this, you don't have clear case control data, but you sort of get this sense that you're getting, the reason you've got mixed data is because it's lower penetrance.
Yeah.
But yeah, I mean, we have this all the time, as I'm sure you do.
And so what do you do in the UK with with the family that I just showed you? Would you guys do the total body MRIs? Would you do the brain MRIs? Like, would you do the whole Lee-Fraumeni syndrome screening? Even though, I mean, the proband had the breast cancer at an early age, but that's about it. And prostate cancer, to my knowledge, is not generally associated with Lee-Fraumeni syndrome.
No, there's not clear. I mean, there's not clear. That clearly, in a sense, a bit of ascertainment bias, isn't it? That slightly may have encouraged the testing or whatever, or just coincidence. Effectively, we're just getting the delivery of the whole body MRI is pretty patchy at the moment. So it's been approved. So in principle, it should be commissioned locally, but that's a bit variable. And I think at the moment, so if you had a report that said pathogenic TP53 variant, and you lived in a part of the country where the whole body MRI was established, you would get it. If you had a report that said VUS, you wouldn't. And I don't think at the moment we've got into kind of nuance of what you do with a TP53 pathogenic variant report that said reduced penetrance. If it's a limited resource, my feeling would be those people should not be a high priority for an aggressive intervention that we are as yet unable to deliver systematically for all the people with the classic TP53 variants.
Right. And I guess here the issue, like, I mean, for our family, it would depend which lab you went to, what result you get, as you saw.
Yeah.
Which I'm thinking, based on the system you guys have, would not be the case, because you would have reached a consensus as this variant was reported out.
I think we hopefully would, because I think we're much better, and we have a nice platform with which to do it. That if something was on the line, people would email round. Not least because if that variant is then, if you do cascading in a family, the sister or the son is going to go to a different genetic center for their predictive test, and the issue is going to come up at that stage. So I think most people, if it's on the line, would preempt that and try and get an agreed consensus of classification up front to make sure they were saying the same thing to the proband that is going to get
right. No, which is great, because that's not what happens here.
Yeah, I know Mark's got his hand up. He may
yeah, I mean, just coming into that conversation, we in the surveillance guidelines we wrote, we can offer we can offer surveillance to the Brazilian people who've got the the low penetrant Brazilian variant. So inference would be that, if, as you said, if this is classified as pathogenic, albeit at lower penetrance, we would offer surveillance, because we just don't know. I think, and this is one variant, one family, so we'd err on the side of caution. But it is tricky, because I mean the ascertainment is very different. I think our criteria for testing P53 are quite high, and so we tend to just identify the families where there is a significant amount of cancer. Because
you're not doing the broader panels, is that right?
We're not doing broader panels. TP53 is in breast cancer, we only do it in under 35s and those where it's HER2 positive, under 35 as well. So it's a fairly tight criteria.
So you would have caught it in this family, because the proband was.
And if it was classed as pathogenic, then we would offer surveillance. We would do the predictive testing and offer surveillance.
Right. but I guess it would just depend whether it was, so would you classify reduced penetrance?
Well, it's either pathogenic or not. Well, the first question is it pathogenic? And if it is, then if it's reduced penetrance or full penetrance,
yeah,
we wouldn't have enough evidence, I think, to make that call, and they would all get surveillance at that point.
No, we're stuck with that too.
Yeah.
But I'm not sure it has enough points to get over the line from the TP53 VSIP guidance as it stands. I don't have it in front of me, but it looks equivocal whether there is enough phenotypic or functional data to get it over the line. Which is why I think would be consistent with some of the classifying labs calling it a VUS.
Yeah, I mean, in the US, we have this, I mean, it's a minority of variants, but it ends up being a huge problem on the patient front. Like when you have the same family with the exact same variant being called out as different variants. And again, we don't have control over it because we have so many testing labs, and we're not always the testing provider, yet we're the ones who see the multiple family members. There's actually a question in the chat that I was looking at. Would there be info in CanVar for a TP53 variant with an N of 1?
So we haven't yet amalgamated the national TP53 data. So in the moment, it's only MMR and BRCA in there. And the TP53 is one of the ones that needs to be done next. So I don't think there's anything particularly in but actually, because it doesn't have any, it doesn't have the Giacomelli and the Cato is only partially functional. So there's no DNE, there's no loss of function assay. So you are pretty much, you're depending largely on the amassed phenotype data, and then the in silico prediction.
But can I just tell you, the phenotype data, I think most of those cases in ClinVar are from my patient's family.
Yeah.
There's only like 5 reports, I think, and 4 of them, or no, 3 of them are ours.
But that's but it's actually that they aren't sort of young sarcomas or kind of super high scoring ones in terms of racking up the points. So you potentially get PM5. I haven't looked exactly at the in silico, but there are other pathogenic variants at that codon, and then PM2 due to absence. So I guess you would need. And then I can't see the Basedell, but so I think you are, but I don't think you've got enough phenotype points to pull it up, even though that just the nature of the cases and families in which it's been observed are quite weak compared to what the VSIP guidance requires.
Yeah, and I'll tell you, it was a tough call because this is an atypical family, probably reduced penetrance. But then with TP53, we generally will test children, right? So we felt like we should move forward with testing the children, even though there were all these question marks about, well, what is the true significance? But I felt like we were stuck. We had to we had to offer the children testing. But again, to the patient, it was more like, hey, yes, this is a TP53 pathogenic or likely pathogenic variant from what many of the labs are calling it, but the risk may not be as high as what you would typically see in the literature.
I think Mark was
just slightly different question. I mean, I think it's been great that you've let Claire show what the capability of of the CanVar program. And I'm just wondering, what, in your opinion, how else can we increase the exposure of this in the US? because most people haven't heard of it.
Yeah, no,
it's quite
I
apparent.
think like
So what else do you think we should be doing if you were us, trying to broaden the user?
Well, I would
profile?
go twofold, right? One of them is, do you want to, like, if you're willing to have users, and you could vet them however you chose to vet people, be involved in the login piece. That piece is really, really useful, right? So again, that would kind of be a carrot. But beyond that, I mean, I think presenting at meetings, right? Like, these would make phenomenal. Mark, I wish that we had kind of thought about it for the ACMG upcoming meeting, but you guys have so much data. You could submit abstracts, and this could very possibly be talks at national meetings. I just don't think people are super familiar with it. The word has not gotten out. Like, you were the first one that had told me about CanVar, but I think national meetings would be a starting point.
Okay.
And we have a lot of ICARE participants, and if you want us to blast anything out, certainly more than happy to do that. Claire, I really, really appreciate you.
No, it's a pleasure. In a sense, also, we've sort of been waiting, and it's a slow project, getting this national data structured and harmonized and so forth, and getting that into CanVar, because that's sort of again another key feature of something we've managed to pull together as a group. So yes, I think you're completely right about just letting people know it's there, but actually maybe kind of going back to the labs and just, I guess, seeing if people feel unhappy.
So I hear this is another way, actually, Mark. Constance Murphy just texted, I would be happy to post the information on the City of Hope portal. We have a working group where all persons trained at City of Hope are invited. That is a massive, massive cohort. That's the meeting that, the City of Hope alums attended. It was at the University of Chicago just a couple of weeks ago. I don't know, Constance might have more, or Connie, sorry, may have more insight into this, but if there may be opportunities to present in their forum at some point, which I think would be tremendous, tremendous reach. So I think there are definitely opportunities.
That's a great suggestion too. If you could, connect us with Connie, that'd be super.
Yes, and I can certainly connect you, Connie. I hope it's okay. I'm just going to connect you with Mark and Claire. And maybe there are, I mean, I would be surprised if they wouldn't. Oh, you know what? Connie,
there we go.
So you guys are now connected.
Excellent. I'll grab that before the meeting closes. Thank you, Connie. We'll make contact and hopefully sort something out. That sounds great.
Yeah, and however we can help, we're happy to e-blast it to our provider partners as well, who weren't able to join the case conference. And maybe we'll do a write-up next time in our ICARE newsletter that we do every 6 months. But I again, hats off to you guys. I don't know how you guys do it. As someone had pointed, I think it was Emily Fossey last time, said, what you guys are able to accomplish in the UK, we wish that we could in some part.
Well, I think, yes, I think there's it swings and roundabouts. And I think, and Mark would probably agree, I think we are, cancer genetics in the UK, we're sort of quietly quite cohesive, and it's nice. And then this CR-UK money we managed to get pays for a few key salaries that just, spare some manpower and time to deliver these projects. So it does, it makes all the difference.
Okay, well, thank you, Claire, for
pleasure.
Presenting. I know we're at time. Just to close out the case conference, our next one is June 2nd. Our guest expert will be Alvaro Montero, who will be talking about BRCA1 VUSs. Claire, anytime you're free or want to join, you're more than welcome.
No. That it's actually quite a good time slot because we're finished for the day. So I may I'll contact you on the side and maybe see if I can get on your invite
we'll add
list.
you. We'll add you to our contact list. Join anytime you want. And we have a slew of guest experts. It's scheduled for the rest of the year. We also have, we always mention the monthly journal roundup, which again is a UK-led resource, again, that really is led by Izzy Turbin. And again, if anyone is interested in getting these notices, just contact us and we're happy to share them. And thank you everyone for attending. And if you have any questions, if there's anything else we can do to facilitate any collaborations or any additional information about CanVar, we're certainly happy to link you to the UK team that's been leading this effort. Amazing resource. So thank you, thank you, thank you.
Brilliant. Thank you very much. I'll just pop the link in the chat for anyone who wants it now.
And if you search CanVar-UK on Google,
it comes up. Yeah.
All right. Thank you, everyone.
Thank
Bye.
you. Thank you very much for having us. Bye-bye.